This lecture is going to be all about mitosis and the process. So in this cell, we have DNA, and DNA is normally composed of chromatin, okay? And I just call chromatin, it just looks like spaghetti, like DNA. But when we are going to division, cells are, DNA are going to become chromosomes. Okay, so chromosomes are made up of chromatin and histones. And then the reason why that chromatin becomes chromosomes is to help compact the DNA for cell division. Now, each species has a characteristic chromosome number. In humans, our diploid number is 46 and our haploid number is 23. So what do these mean? Diploid, the prefix di means two. Okay, so cells have two of each type of chromosome. You get one from your mom, one from your dad. So we have 23 pairs for a total of 46 chromosomes. So you might see sometimes people say 2N in humans is 46. N just stands for number of chromosomes. So we have 2 times the number of chromosomes, so 23 pairs. And then haploid, think half. So they have half the number, and this is going to be an egg and sperm. So our N number is 23. And this will be important later on. Now, in a regular cell, this is just an example. So our 2N number is 4. We have 46. That's our 2N number. But in this just example, so a normal cell, this has 4, okay? So your chromatin becomes chromosomes. Our number is still 4. And then the process of mitosis, we still have the same number. So mitosis, we are not multiplying, we are not dividing in mitosis. 2N cell is going to create another 2N cell. So our chromosome number is going to stay constant, and this is going to be important because we want all our cells to look alike. We want skin cells to look like skin cells. We want stomach cells to look like stomach cells. So it's really important. Now, when these chromosomes get formed, we have one chromatid. This is from the first video, okay? We get replicated during the S phase and interphase. So this is not mitosis, this is interphase. Okay, so we have 2 sister chromatids that are held together at the centromere. And then when cells divide through mitosis, they go from chromosomes to chromatids. So mitosis is a nuclear division that forms 2 daughter nuclei with the same number and same kinds of chromosomes. The daughter cell are the same as the parent cell. So let's go ahead and talk about the process of mitosis. So for me, the easiest way to remember the steps of mitosis is to remember the acronym PMAT. Okay, if you remember PMAT, you'll be able to remember the order of mitosis. So the first stage in mitosis is prophase. So in prophase, our nuclear membrane is going to disappear. Chromatin is going to become chromosomes. Okay, and then we're going to have these things called spindle fibers. They're going to be important in a second, are going to attach to our chromosomes. You have like this subphase called prometaphase. You don't really need to know about this. This is just an addition to prophase. So right here we have prophase, early prophase. So our nuclear membrane is starting to go apart. Still prophase. Our chromosomes are starting to form, right? So we go from chromatin to chromosomes. And then in prometaphase, we have these things called spindle fibers are going to be attaching to our chromosomes. So that is all prophase. You don't have to know early prophase, prometaphase. Just know this is all prophase where this occurs. Okay, so next, we're still doing PMAT. That was prophase. Next is metaphase. For metaphase, I want you to think middle. So in metaphase, our chromosomes are going to line up in the middle. You'll see them something like this. Then we have anaphase. For anaphase, I want you to think apart. So our chromosomes that were nice little X's are going to pull apart into chromatids. So anaphase chromosomes go back to chromatids where they were in prophase. And then last in our PMAT is T telophase, think 2. So now we are going to have almost 2 complete cells. So our nuclear membrane is going to reform, and our chromosomes are going to start to go back to chromatin. So metaphase, they line up in the middle. We still have these spindle fibers attached. Anaphase, our spindle fibers are now going to pull the chromatids apart. Telophase, we have 2 nuclei, right? We don't have 2 complete cells yet because we still have one more step that has to happen. But this was a metaphase, anaphase, telophase. Now, mitosis in plant cells is a lot similar to animal cells. So there's not too much difference. One difference is you don't have centrioles, which are these organelles that help pull the chromatids apart. So plant cells do not have centrioles. So again, prophase starts to disintegrate. We start seeing chromosomes form. Metaphase line up in the middle. Anaphase are still pulling apart. Telophase, you have 2 nuclei. What is different, however, is the last step of cell division, which is called cytokinesis. So cytokinesis literally means cytoplasm split. So we're splitting our cytoplasm. So we went from having these 2 nuclei, now we're going to have 2 cells. So in animal cells, we have what is called the cleavage furrow form. So we're going to have, it looks kind of like this, okay? A contractile ring is going to form right here, and it just pinches it apart. It's kind of like if you have Play-Doh and you pull Play-Doh apart into 2 different pieces. That's what happens. It's called a cleavage furrow. Okay, so here we have 2 cells, cleavage furrow, a ring. Think of it just being like a rubber band, just pops it in half. Okay, and then eventually they will separate. That's in animal cells. Now in plant cells, you have what's called a cell plate forming. So remember, plant cells have cell walls, animal cells do not. So in plant cells, you're going to have a cell wall form in between those 2 nuclei. Okay, so right here we have a cell plate forming that is going to go between cell 1 and cell 2. So that is mitosis right there.